Students, how are you all? Let's meet once again with another unit of organic chemistry, that is biomolecules. So, let us start with the unit biomolecules. This is your mentor Shweta Soni. Now, to explain what are biomolecules, these are the naturally occurring substances or these molecules in the biological systems. Biological system, that means in the organisms, and hence they are termed as biomolecules. We will be studying about different biomolecules in this chapter. That is, we will be studying about carbohydrates, proteins, enzymes, vitamins, etc., and nucleic acids. So, let us in detail study about the carbohydrates, which is the first category of biomolecules. If we discuss what are carbohydrates, these are optically active polyhydroxyaldehydes or ketones. When I say optically active, what do I mean? Optically active means they have ability to rotate the plane polarized light. Polyhydroxy, when I say, that means they contain more than one OH or hydroxy group. Aldehyde or ketone, that means they contain the main functional group as either the aldehyde or the ketone. So carbohydrates are optically active polyhydroxyaldehyde or ketones, or the compounds which produce such units on hydrolysis. For example, we will be studying about glucose, sucrose, cellulose, starch. All these are carbohydrates, where you can see glucose is a monosaccharide, sucrose is a disaccharide, cellulose and starch are polysaccharides. Now, how can these carbohydrates be classified? They can be classified on the basis of the number of hydrolysis products they yield. For example, if we talk of monosaccharide, these are the simplest carbohydrate which cannot be hydrolyzed further, and they are termed as monosaccharide. They contain only one unit. So they cannot be hydrolyzed. For example, glucose and fructose. Remember, glucose contains aldehyde functional group and fructose contains the ketonic functional group. Oligosaccharide is another category of carbohydrates which yield 2 to 10 monosaccharide units on hydrolysis. For example, sucrose, maltose, lactose, all these three are the disaccharides. Disaccharide, they are formed by two molecules of monosaccharides. Raffinose, stachyose are another examples of oligosaccharides. If you see polysaccharide, they are those carbohydrates which yield a large number of monosaccharides on hydrolysis. We will be studying about the three polysaccharides in detail in this chapter, that is starch, glycogen and cellulose. Now let us see what is the importance of carbohydrates. Carbohydrates, if it comes as a 2-mark question, you have to write at least 4 points related to importance of carbohydrates. So all of us know that carbohydrates are energy reservoirs. That means they act as biofuel and provide energy. They can act as storage molecules, for example, starch in plants and glycogen in animals. If you talk of nucleic acids, DNA and RNA, both of them contain a sugar. That is, DNA contains 2-deoxyribose sugar, whereas RNA contains ribose sugar. So these sugars, they are also carbohydrates which are present in nucleic acids, DNA and RNA. Cellulose, which is a polysaccharide, acts as a structural material of cell walls of bacteria and plants. They also provide raw materials for various industries like textile, paper, lacquer and breweries. So these are the important functions, and remember these functions, 4 points, it can come as a 2-mark question. Now let us see another classification of carbohydrates, which is on the basis of the reducing properties. Whether they are able to reduce the Tollens reagent or Fehling solution, or whether they are not able to reduce. So depending upon that fact, if they are able to reduce the Tollens or the Fehling solution, they are reducing sugars, and they are those carbohydrates which contain a free aldehyde or a ketonic functional group, and they are readily able to reduce Tollens or Fehling solution. For example, all monosaccharides will come in this category, and the disaccharides will also come in this category, remember, except sucrose. We can take example of maltose, lactose, they are reducing sugars, but remember sucrose is a non-reducing sugar. That we will discuss why. Non-reducing sugars are those which do not have a free aldehyde or ketonic group and do not reduce the Fehling or the Tollens reagent. And the example is sucrose. Let us see how can we prepare sucrose. Sucrose, preparation of glucose. So we will start with the monosaccharide glucose. We can prepare it from sucrose. Sucrose, when subjected to acidic hydrolysis, will give us one molecule of glucose and one molecule of fructose. Now, another method to prepare is by the acidic hydrolysis of starch. We can prepare it by the acidic hydrolysis of starch. When starch is subjected to acidic hydrolysis, it breaks down to form from starch we can prepare glucose by acidic hydrolysis. When starch is subjected to acidic hydrolysis at 393 Kelvin and 223 atm, we get n number of glucose molecules because starch is a polysaccharide. Now, what is the structure of glucose like? Structure of glucose contains 6 carbon atoms. So we say it is a hexose sugar. It contains one aldehydic group, it contains one primary alcoholic group, and it contains 4 secondary alcoholic group. So remember, we call this glucose as aldohexose sugar. Why so? Because it is containing aldehydic functional group and it is containing 6 carbons, so it comes in the category of aldohexose sugar. Now just look at this carbon number 5, because this is 1, 2, 3, 4 and 5. Look at this carbon number 5 and let us compare the configuration of the OH group on this carbon 5 of glucose with respect to the reference glyceraldehyde. This is D-glyceraldehyde having OH on the second carbon on the right side. This is L-glyceraldehyde having OH on the on the middle carbon on the left side. So with reference to the glyceraldehyde, if the OH on carbon number 5 is on the right side, it is D-glucose. But if it is on the left side on carbon number 5, it is said to be L-glucose. But remember this positive sign here, that this positive in brackets or this negative in brackets does not mean capital D or capital L. It is actually the optical rotation. For positive, that means the optical rotation is on the right side, dextrorotatory. But for negative, it is on the left side, it is levorotatory. This is in no case related to the capital D and capital L configurations of glucose. So this is all about the D plus glucose and the D plus glucose and L minus glucose. Now, if you just see the cyclic structure of glucose, how can we draw the cyclic structure of glucose? To explain the cyclic structure, just see carbon number 5 of the glucose, which is having OH group on the right side and H on the left side. When from this OH group, the hydrogen gets attached to the carbonyl oxygen of the aldehyde group, then there is a hemiacetal linkage which is formed between C1 of glucose with C5 of glucose. And this hemiacetal linkage basically is formed between C1 and C5, and these are actually the Fischer projection structures of glucose. Now, here also the hemiacetal linkage is formed. Now we have to focus on carbon number 1. Look at carbon number 1 in this structure, alpha D plus glucose. You can see OH is lying on the right side. If OH lies on the right side at carbon 1, it is alpha D plus glucose. If it lies on the left side at carbon 1, it is beta D plus glucose. Remember, alpha and beta D plus glucose are anomers of each other, which differ with respect to the configuration of the OH on carbon number 1, and this carbon 1 is called the anomeric carbon. This is called the anomeric carbon. So these are the cyclic structures, and these are the two anomers of glucose. Alpha D plus glucose having OH on the right side at the anomeric, that is the C1 carbon, and beta D plus glucose having OH on the left side of the carbon number 1. These are the Fisher projection formulas of the cyclic structures of the glucose. Now let us see how can we draw the Haworth or the Pyrenees ring structures of glucose. So to transform the Fisher projection to the Pyrenees ring structure, we have to keep this in mind that right means bottom. Whichever group is present on the right side has to be placed at the bottom. So just have a look here that there is a linkage, that is a hemi-acetal linkage, which is linking C1 with C5, and on carbon number 1 you can see here this is the alpha anomer Haworth structure. You can see that at carbon number 1 OH is on the right side, hence placed at the bottom. 2 OH is on the right side, hence placed at the bottom. 3 H is on the right side, hence placed at the bottom. 4 OH is on the right side, hence placed at the bottom. So these are the Pyrenees ring structure or the Haworth structure of glucose, and Fisher projection structures can be transformed to Haworth structure by keeping only this in mind that right means bottom. Whatever group of atoms is placed on the right side in Fisher projection has to be placed at the bottom in the Haworth structure. So this is about the Haworth structures of glucose. And now we come to the next, and that is the chemical reactions of glucose, where we will learn about, let us learn about the structure of glucose. The structure of the reactions of glucose. So we just have a look at this flow chart which tells us about the reactions of glucose. This is the condensed formula for glucose, and when glucose is treated with a reducing agent HI, then it converts into N-hexane, and this confirms the presence of straight chain in glucose. Let us see what will happen when it is treated with bromine water. Remember, bromine water is a mild oxidizing agent which will convert or oxidize only the CHO group, aldehydic group, into carboxylic acid. The primary alcoholic group will remain as such, resulting in the formation of gluconic acid. So this reaction confirms the presence of the aldehydic functional group. Next is the reaction with acetic anhydride. With acetic anhydride, you can see that glucose readily reacts to form. Now here what will happen? Acetylation will happen. Acetylation means the replacement of active hydrogen of the alcoholic group by the CH3CO group, acetyl group. And you can see it will form a penta-acetate of glucose, meaning that it confirms the presence of 5 OH groups in the structure of glucose. Now, let us see what will happen when it reacts with hydroxylamine. So like we have studied in the chapter carbonyl compounds, that when any carbonyl compound is treated with hydroxylamine, condensation reaction occurs. The carbonyl oxygen of the aldehyde along with the 2 hydrogen atoms of hydroxylamine are lost in form of water, resulting in the formation of compounds called oxines. So this is a glucose oxine which is formed. Now let us see the reaction with HNO3, which is a strong oxidizing agent. Now with mild oxidizing agent, what did we observe with bromine water? That only CHO group was oxidizing to COOH, indicating the presence of aldehydic functional group. But here you can see with the strong oxidizing agent, not only CHO group gets oxidized to COOH, but also the primary CH2OH alcoholic group also changes to COOH, resulting in the formation of saccharic acid. So this reaction confirms the presence of primary alcoholic group. Next is the reaction with HCN, and when any aldehyde or ketone, that is carbonyl compound, reacts with HCN, they form cyanohydrins. And glucose also is forming cyanohydrins, where you can see that to the oxygen, carbonyl oxygen, the hydrogen is attached, and to the carbon, positively charged carbon, the CN- group is attached. resulting in the formation of glucose cyanohydrins, and this confirms the presence of carbonyl functional group in the structure of glucose. So remember, all these reactions are very, very important. They are very easy if we have thoroughly learned about the unit, that is the aldehyde ketone. We can easily memorize these reactions. Now we move on to the structure of fructose. The fructose is also a monosaccharide, but unlike glucose, which is an aldohexose sugar, it is a ketohexose sugar, meaning that it contains 6 carbon atoms along with the presence of ketonic functional group. But remember, it has the same formula as that of glucose, C6H12O6, because glucose and fructose, we can say, are functional group isomers of each other. It belongs to D series and it is a levorotatory compound. Why? Because its optical rotation, or the direction of rotation of plane polarized light, is towards the left side with an angle of the optical angle of minus 92.5 degrees. Remember. So this is the structure of the fructose, and these are the cyclic structures rather of the fructose, where in glucose you saw the one carbon was linking with the carbon number 5 via hemiacetal linkage. Here the number 2 carbon, which was actually the functional group carbon, is linking to number 5 via a hemiacetal linkage. So again you have to focus on this carbon number 2, which is called the anomeric carbon. If it has OH on the right side, it is alpha D minus fructose. But if it has OH on the left side, it is the beta D minus fructose. So now we will learn, these are the Fischer projection cyclic structures of the fructose. Now how can we transform them into Haworth structures? So we will be making the furanose ring structure, where you can see number 2 and number 5 carbon atoms will be linked via oxygen. This is a hemiacetal linkage between 2 and 5, and here number 2 is called the anomeric carbon. So these are the cyclic structure. To draw the cyclic structure, again the same thing has to be kept in mind, that right means bottom. So at number 2, just see that OH is placed on the right side in alpha. So in alpha, OH has to be placed at the bottom. On the other hand, if you have to draw beta anomer, then you have to place OH on the top. So these are the Haworth ring structures for the fructose, and this is all about the monosaccharide, that is the glucose and the fructose. Now we will learn about the disaccharides. So we will be learning about the 3 disaccharides. So what are the 3 disaccharides? Sucrose, we will learn about sucrose, we will learn about maltose, and we will learn about lactose. So let us start with what are disaccharides. It is a category of belonging to oligosaccharides, where oligosaccharides, you know, on hydrolysis yield 2 to 10 molecules of monosaccharide. Now, disaccharide are those carbohydrates which on hydrolysis give 2 units of monosaccharides. Disaccharides are soluble in water and exist as crystalline solids. Sucrose, maltose and lactose are examples of disaccharide. If we talk of sucrose disaccharide, this on hydrolysis, we had learnt earlier also, gives 2 monosaccharide. One is the glucose and another is the fructose. So a question comes, if relevant to this, that why is sucrose called invert sugar? Now just see, the optical angle of the sucrose is plus 66.5. But when it is hydrolyzed, it results in the formation of glucose with an optical angle of plus 52.5 and fructose with an optical angle of minus 92.5. So you can see that after hydrolysis, the sucrose, which is originally dextrorotatory, changes to levorotatory. That means inverts its direction of rotation. Hence, this is called invert sugar. This question is a very, very important question which often comes as 1 mark reasoning question in your board examination. Let us now learn about the ring structure of sucrose. Ring structure of disaccharide sucrose, where you have already learned that sucrose, being a disaccharide, is formed by the combination of alpha D plus glucose and beta D minus fructose. So here you can see that C1 of glucose links to C2 of fructose via a glycosidic bond. These linkages between the two monosaccharides in a disaccharide are called the glycosidic linkages. So you can see that this glycosidic linkage is formed between the C1 of glucose with C2 of fructose, and hence none of the functional group is left free to exhibit the reducing properties or to reduce the Tollens reagent or the Fehling solution. Hence we say sucrose is a non-reducing sugar. Let's talk of the second disaccharide, which is maltose, present in malt and hence called malt sugar. The monomers are, it is made up of two molecules of glucose, where you can see this is one molecule of alpha D glucose and this is another molecule of alpha D plus glucose, and C1 of one molecule forms a glycosidic linkage with C4 of adjacent glucose unit, and hence this is actually a reducing sugar because it will show reducing properties. It can reduce Tollens or Fehling solution, hence it is known as reducing sugar. So maltose is formed by a combination of two molecules of alpha D plus glucose, where C1 of one molecule links to C4 of another molecule via a glycosidic linkage. Now let us see the third, and the third one is known as the fructose. The